Is cancer caused by intrinsic or extrinsic factors, and does it matter? At a Cancer Research UK symposium in Oxford, keynote speaker Richard Gilbertson explained what can be learnt from the debate.
So if we think about cancer, it's like many diseases. You can think about the underlying factors that contribute to it in two ways. One is the host, that's the organ themselves, that's the person themselves, that's the tissue that will generate the cancer. But then there's the outside elements that will impinge on that. And when it comes to cancer, people think about, for example, smoking, so cigarette smoke. So it's this interplay, complex interplay between the organ itself that's going to generate the cancer, the cells in that organ, but also the external factors which change the microenvironment that come into the cell and have an effect. And it's that interplay which contributes to cancer.
But the recent suggestion that cancer was intrinsically caused has sparked some debate.
Yeah, so actually within the last 2 or 3 years there's been a sort of debate raging around the so-called bad luck hypothesis, which is an interesting idea, which was proposed because some tissues have very high burdens of stem cells, lots of cells that are primitive, that generate the organ, that are able to proliferate for the life of the organ. And so the idea is that because they will just spontaneously get mutations, and because they constantly proliferate, then that in itself is enough to cause cancer, and therefore the implication is you can't prevent cancer in those organs because it's just bad luck. We happen to have those cells, they happen to get mutations, and so you will inevitably get cancer. So what we've been working on is looking at how true that is, because that data has been generated by taking existing cancer incidence data and existing published stem cell data and pushing the two together in mathematical models. And actually the mathematical models have come up with conflicting results. So we've been using laboratory systems to actually test that actually experimentally. You can look at how normal cells function in a tissue. What are their properties? Are they cells that just sit there and do the function of a tissue but they don't divide, they don't grow? Or are these the stem cells which generate the tissue, which are actually proliferative? And if you have that function characterized, you can then ask, are they susceptible to make tumours or are they not susceptible to make tumours? And then you can ask things like, what does the intrinsic environment do to that? So if you bring something in that would damage those cells and make them proliferate more, does that then make them more likely to make cancer? So it's that we've been piecing together.
You've particularly focused on PROM cells.
Yes, a number of years ago there was a gene called CD133 or promin1 that was discovered as a marker of stem cells and also thought to be at the heart of most cancer stem cells, which is a fraction of cells that are thought to be particularly tumorigenic. The only reason we used it is simply we wanted to target a specific defined population of cells. There's nothing special about promin1 or CD133 in our system. It just allowed us to actually genetically target a defined population of cells.
And you've looked across different ages and different tissues.
Yes. And so if you look across different tissues at different ages, you can see the general function is that there is a relationship between the generative capacity of cells, how much they're able to actually proliferate and make tissues, and whether they're not susceptible to cancer. And also we've been able to show in general terms that part of that generative capacity is provoked by extrinsic damage, by damage to those cells. So then it starts to piece together this complex dance in a tissue between damage, the response to repair the organ, and then the presence of a mutation. And it's that perfect storm that comes together to generate tumours. What's critically different, however, from the debate that's gone on is that we don't think it's purely a bad luck aspect. The notion that you can't prevent these tumours because they're just simply bad luck, we don't think is true. We think the stem cell function is important, but that can be provoked by extrinsic factors. And so if you are exposed to carcinogens, which can include cigarette smoke, alcohol, etc., then if that provokes an increased stem cell capacity within your organ because it's trying to repair the tissue, then that will increase your risk of cancer.
It sounds like you've gone a long way round to come back to the idea you first thought of.
Exactly, and that's actually the story that's the history of science generally, actually, and there's nothing new under the sun. I mean, the notion that stem cells could be at the seat of cancer is decades old. It's not a new idea. I think what's different now is we have the technologies, the laboratory tools accessible to test those hypotheses and provide robust data in support of what was maybe a very old idea. And often old ideas are often the best ideas, and they've stood the test of time, they've stood the test of debate, and all we're really doing is coming along maybe and testing somebody else's idea with novel technology.
What practical things have you learnt about stem cells?
What has been exciting is we know if you sequence genes, if you look at the genetic code in tumours, we know that about 20 to 40% of genes, we'll know what their function is. We'll know that they are so-called oncogenes, drivers of cancers, or tumour suppressors, genes that are brakes that would normally try and stop a cancer from happening. But 80% of genes that are often mutated, we don't know what they do. And this system has been enabling us to predict what those genes do, because we can look at their expression in the stem cells that give rise to those tumours. And based on the pattern of expression, we can start to infer whether they're likely to function as oncogenes or whether they're likely to function as tumour suppressors.
So what next in research?
For example, there's a huge wave at the moment in immuno-oncology, using the immune system to target cancers, and the fact that the immune system might be disrupted in cancers. What has been particularly exciting is coming at this with a stem cell background, completely agnostic to that. What emerges is a very strong immune story from those genes. And so I think that's very exciting when you start to see fields converge, and you start to see answers that other people are coming up with from different fields. And that's when it starts to feel real. It starts to have a sense of robustness about the data.